Hello, my name's Neil Spooner, and I'm the chair of the Patient-Centric Sampling Interest Group, and I'd like to welcome you to today's webinar, Analytical Mass Spectrometry Workflow for VAMs and its applications in cardiology and beyond. And I'd very much like to welcome our speaker today, Jenny Van Eyck from Cedars-Sinai Medical Center in the USA. Before I hand over to Jenny, just like to thank our partners and sponsors whose support of the Patient-Centric Sampling Interest Group means that we can bring you these monthly webinars and we can have our other online events and working groups. So thank you very, very much to our sponsors. All of you in the audience, please use the chat function to submit your questions. Jenny's going to leave some time at the end to answer your questions. So please just submit them through the chat function. This event is being recorded and will be available through our website and through the PCSIG YouTube channel in the coming days. We'll make an announcement to the LinkedIn community when it's available. So with no further ado, I'd like to hand over to you, Jenny, and thank you very much.
Well, thank you for that nice introduction. So good morning or good afternoon or good evening to everyone who's on this call. And it's my pleasure to give a talk, an update really of the work that we've been doing using remote blood sampling devices and its applications within the context to precision medicine. So there really is, I think, a need to rethink how we do studies in order to obtain information that will be longitudinal, will reduce barriers to access to biological or biochemical data around the around the individual, and also to just explore rare diseases and natural history. So we do this really in context to an understanding that diseases are complex and that an individual can have the same clinical phenotype, but yet what's driving that clinical phenotype can actually be due to multiple and different biological processes that are going on in an individual. And often, unless it's an acute injury or a disease like that, usually they're very complicated. And understanding and tracking over time can help us be able to diagnose someone, determine, their response to therapy, and also give insights, hopefully, around the mechanisms that's driving those different pathways. So we adopted the use of remote sampling devices, and the one we most commonly use is the Meter device from Neoterix and Trojan now. We like this one because it's volumetric. So essentially, if you fill and wick up the end of the device, you would get close to 10 microliters or 20 microliters, depending on which one you've chosen. We often use that for our own lab. We often use the 10 microliters, and we usually ask people to fill twice, so 2 different tips. We have gone out to 4, but we find fatigue, let's say, in our patients or in our subjects who are doing them. And so we find consistency with having 2 devices per time point being done. And of course, the advantage of remote sampling and dried blood is that one is the patient or the individual can do it themselves. They can do it anytime, doesn't matter where they are, and really under any circumstances. It's also really nice, at least in the United States, once the blood's dried, it's considered no longer to be a biohazard, so it can be mailed to us just using standard mailing devices. It can be stored at room temperature, and I'll show you some data to support that in a few minutes. And so it really allows a lot of flexibility for the individual, and then to get the sample back to a core lab or to our lab, whichever. We primarily use mass spectrometry to analyze the content of those blood. And that's because we can digest the proteins which are in that blood sample within the actual tip itself, and we get great recovery, and I'll show you some of that data also in a few minutes. And I'll go a little bit into mass spectrometry and the analytical parts of why that makes it a good measure for proteins. But we can either do discovery, which means we're trying to see as many proteins quantitatively as possible, or targeted assays where we're really focused in on a subset of proteins and have made very accurate and precise measurements using that. And we, of course, I'll have data to show that. So again, the one of the advantages of using devices like the Meter device, or being able to have home, individual sampling, is that you can then track over time, right? And this ability to track over time allows you to watch not just the dynamics of and release and clearance kinetics of a biomarker or biomarkers, but actually allow you to try to understand natural reflection in the plasma or blood across time and being able to track that. And there's lots of advantages to it. And so we try to insert these types of measurements in cohorts where otherwise just getting plasma is not really conducive. So I'm going to talk about the very first study we ever did that actually involved a cohort. It was funded by the California Institute for Advancement in Precision Medicine. It's a state funding source. It was a competitive grant, and there was 3 PIs, myself, Noelle Berry-Mertz, who's a really remarkable cardiologist working really in women's health, and Brennan Spiegel, who does a lot of using devices and things like that to be able to do not biomarkers, but things like Fitbits and virtual glasses and things like that to be able to track disease progression over time. And so we came up with the idea, this was like in 2016, that maybe we could also include circulating biomarkers as a way to explain fluctuations or things we're seeing in questionnaires or seeing in Fitbit measurements that were indicative of a specific disease setting. The disease that we decided to track, we wanted to, because it was the first time we were going to deploy remote sampling devices, a kind of highly motivated group. And so Noelle works with many individuals, but out of her group, we did individuals who had already had a cardiac event. So they are mid-risk individuals. So they have about 20% would have a secondary event within a year. So 15 to 20% would have rehospitalization, they'd have arrhythmia, they would have death, or they would have been hospitalized for heart failure and then as a rehospitalization. So that's called MACE. So it's, and then so we wanted to be tracking these individuals over that time point. And at Cedars, what we do is when you come in with having already had your first event, you do go into rehab. So they're coming in for the X number of months every week for their rehab. So we were also able to interact with them if they were having troubles doing their actual remote sampling devices, or to be able to answer questions and try to work with that to try to get to a point where we knew that it was going to be a really good uptake. And I think we managed to achieve that. So the study design was they're going to be, everyone will have a Fitbit, it will be continuous use. That we had patient-reported outcomes of several different studies that they did, and that was done weekly to be reporting, did they sleep well, fatigue levels, things like that. And then we did a baseline after when they first came out, we had a plasma sample. We taught them how to do the finger prick, showed them. And then we had them collected 30 days and 60 days. About a few days before they were to do that, we sent them an email to remind them, and then on the exit, and then we did an extension as well. And then we were looking for 1 year outcome. But the first study I'll talk about is really taking the baseline and actually, because we're also measuring cardiac troponin I and BNP and CRP as kind of more at the clinical level, we were able to correlate some of the proteomes that we were seeing in the blood sample to these more classical circulating biomarkers. So again, as I said, they were highly motivated. They were coming in through the cardiac rehab program. The individuals who run that have incredible, great relationships with the patients coming through. And so there was a great buy-in. And behind that, on our side, we had already built out a technical pipeline that had automated, and I'm going to show you a little bit about that, automated sample preparation for mass spectrometry, where we have to reduce and alkylate cysteines. We have to be able to digest it with trypsin really efficiently. We have to be able to extract those peptides out, then analyze them on a mass spectrometer. And what you can see is a tracing below, which is a mass spectrometry peak area. And each peptide peak is actually quantitative in this peak area underneath it. And we'll talk a little bit more about how that's doing. And again, we're going to be measuring this against cardiac troponin I, BNP and CRP, as well as the PROs and the virtual Fitbit measurements. So we had, as I mentioned, already started to work on automation. Phil, do you want to just run the video so people can see it working, and then I'll talk about what we've done? That are able up on an i7 system out of Beckman Coulter. We originally started on the Biomech NPX system, and we worked out all of the steps to make an efficient hands-off process where we, as I said, we reduce the proteins, alkylate them, quench it, have to do a dilution, we do a digestion, we do desalting, and then it automatically goes up into our platform for data analysis and data storage. And from that, we had this already working for plasma, for cells, for all kinds of things. And so we ended up putting on the dried blood. And if we go to the next slide, essentially what we do is we take off that very tip of the meter device and stick it into the wells, and it just goes through it. And then we make sure we had good recovery. If we're doing dried blood spots, it's done very similarly. There's just a first step that we have to do off the little tissue piece or filter paper piece. And again, this is just a way to really have high quality data all the time by controlling it. Along the ways, we also, because we're going to be running a targeted assay, we're going to be running internal standards of the peptides that represent those proteins. So again, because this is digested peptides, we use selected peptides which have an amino acid sequence that's unique to our target protein, one that works very well in mass spectrometry, has good sensitivity, robustness across all kinds of different things that can happen in plasma or blood. And then we include the equivalent peptide, but we've labeled it with a slightly heavier isotope. And so it's going to elute at the same time, but be a different weight. And all of the peptides will have this different weight. So essentially, we're measuring 2 peptides for every single endogenous peptide, the endogenous plus the internal standard. So we're going to explore first a multiplex assay. It's targeted. It's the 11 apolipoproteins. We also include hemoglobin and albumin. So it's actually 13 proteins, plus we have another control, which is beta-galactosidase, and we add that at the beginning of the whole sample processing as a processing control. So we add in beta-gal. It should not be in plasma or in the remote sampling devices. We then are tracking at the very end. We add the SIL peptides right before it's going to be dried down and going on to the mass spectrometer. And so we have peptides for the 9 apolipoproteins we're measuring, hemoglobin, albumin, beta-gal. There's 2 peptides per protein. And because this is done on a triple quad, we're actually not just measuring the peptide, we measure what's called the transitions. And that is the breakdown products of the peptide that provide us with sequence level data. And I'll illustrate that a little bit later. But the concept is that we have 2 peptides per protein. We have a quantifier peptide and a qualifier peptide. The quantifier peptide is usually the most sensitive, the most robust peptide, and that peptide is the one that we actually use to quantify. So we do the peak area under it. The second peptide is the qualifier. It's there really to make sure that you're measuring the right protein, right? Taking after kind of immunoassays where you have 2 antibodies to 2 different regions of the protein. The presence of both epitopes really helps confirm the specificity of the assay. So in mass spectrometry, we usually have 2 peptides to also make sure we have that specificity. But because one peptide usually is better, meaning more sensitive, more robust, that's the one we quantify off. But you'll see later, I'm going to be measuring both peptides and asking the question, if for some reason a quantifier peptide does not work, can we substitute the qualifier? The other thing we did in this project, because again, it was at the first time, we ran both tips. So we have duplicate runs on every single sample. And again, addressing the issue, if you have 2 meter devices, are they actually equivalent? Like, are we going to get the same data? And can we really move eventually into doing just 1 tip and not having to do 2 tips, for example? These are the questions that we were trying to answer with this. So this is just to show you a summary. We have, of course, data. The paper's been published for every single one of the peptides that we're analyzing. And we were worried about short-term and long-term stability of the assay. So just like you were doing a plasma assay, you'd look for digestion efficiency between plasma versus meter devices, between the peptides. Are we really getting as the best tryptic digestion? Are we getting the best extraction recovery from the tips? And what's the stability of the devices? Like, are they stable, those particular peptides we're going to measure? We did it for all of them. And what we essentially do now, we know that for most of the proteins, and we now do discovery where we're looking at, thousands of peptides, that it's incredibly stable. Not every peptide, but the vast majority are stable. So once the meter tips are dried, they're very stable. They can be kept at room temperature. But what we did, and if you go back to the paper, you'll see we did all different kinds of temperature, seeing whether or not we were getting an effect at all on the stability of peptides that we're going to be measuring across time, to make sure that if we get our data, we can actually trust it. So all of that was done behind the scenes. But one of the first things we learned in the trial is that, because we showed them how to do it when they came into the rehab, and then we gave them the kits to go home with, and then we got the first ones back. And what we noticed is not all of them were perfect. They weren't just the tips. And you can see in the photograph on the one side, we got all kinds of overfilling, we got underfilling, we got partial filling, we got wet tips. And so essentially, and you can see at the bottom in the bar graph, as we got more and more tips, after the first time through, we actually made photographs and improved our instructions to this cohort to be able to do it. And what we're trying to show is in the kind of the neon green are the tips that were good. And in the beginning, we were only getting 60% of all the tips to be good. But as you can see, as the end, people got to understand how to do it. The instructions were better. We could do more things. So it worked quite well. And by the end, people had really gotten the hang of it. And we've now used this method for many, 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 many cohorts. And essentially we get, really 95% or better of the tips being well filled, dried, and appropriate. And that was really just not us teaching them, meaning you talking one-on-one, but actually just the information that went along with it. And in fact, in the graph below it, I like this one because it shows that we were comparing across time how many people were using their Fitbits all the time, how many people were answering their questionnaires all the time, and how many people were actually taking their meter devices. And we got really good compliance. It was around 95% of people complied on all time points, and it was equal to that of the Fitbit or the PRO. So for this cohort, again, remember they're mid-risk. They know that they're at risk of having something, but not, mid-risk is mid-risk. And they were really considered taking their blood sample to be as equal as anything else, and they did it. So that's a good thing to know. So when we're actually, of course, testing the assay, we have a lot of QC samples we're running at the same time. And this is just looking at one of the peptides that we're measuring over time. In all the QC samples that we were doing. And we had lots of batches, so they're 96 well plates, and we had a lot of those that we had to analyze to get through this cohort. And you can see that it's fluctuating about normal. And what you can do is the peak area is what you're measuring, and we have different transitions. You can see the breakdown products, and that really is a reflection of the amino acid sequence of the peptide. But when you have in the internal standard, right, the heavy labeled peptide equivalent, you can then you can normalize to area ratio. So it's the ratio of the endogenous over the external control, and that gives you a normalization, and that should hover around 1. And this worked incredibly well to normalize any small deviations going under it. And that's really how these targeted assays are done. We do peak area ratio. So in a paper where we wanted just to get the data out, it ended up in Circulation. We compared the concentration, the initial concentration, and asked the question, we measure in these individuals cardiac troponin I, which is really a heart injury marker, BNP, which is really about hemodynamics, or CRP for inflammation. And did any of these proteins, the apolipoproteins, were able to be reflective of that state? And there was, not to our surprise, but some of them were, like APO2A actually had good p-value across all of these 200. It was 198 individuals. We had 2 individuals that left the study, so it was 198 individuals. But I also just wanted to point out the amount of chemical data, analytical concentration-dependent data that one's obtained. So as I mentioned, it's an 11 protein assay. We're measuring 22 endogenous peptides, But of course, we also have the cells. So we have twice that many. We have 44 peptides that we're measuring. And because this is MRM, we're measuring the transition, the breakdown products that's part of the sequence. So we're actually measuring 66 transitions. And we did it in 198 patients. We did 2 tips per person. We did only 1 time point. And that's actually, to get this data that's in this little chart, was 52,000 data points that was actually analyzed to obtain that data. So compare that to an immunoassay where you would have had 1 assay, right? You'd have 11 proteins, so 11 different assays. You would have done it in duplicates, so 22 runs, and you would have done that on 198 individuals. So you'd be just under 400 data points. So this is the quality difference between the analytical quality difference between something like an immunoassay and something like mass spectrometry. We have a lot of redundancy in the data that you do. And one of the questions early on was, do you really have to measure the 52,000 data points all the time? Or is there, like, do we just have to measure 1 peptide all the time? And if it doesn't look like it's meeting QC, can we flip to the qualifier? And we did that, and it's a paper also that was published where we went through all of that data and really asked the question, how many times is really the quantifier good? How many times does the quantifier peptide actually fail, and can the qualifier do it? And then the other question was, do we have to measure it in both tips, or can we just use 1 tip? And if that 1 tip doesn't meet QC, can we flip to the second one? So this is a heavy paper to read, just to really answer those simple questions. And you can go through all the decisions and cutoffs that we decided to be able to do. But the bottom line, which is shown in this complicated graph, but it's really not that complicated. It's something like 95% of the tips worked, and it worked on the quantifier. They were all fine. The majority of the time, the data is great on single peptide, single tip use. However, the times when it failed, of those that did fail, which was very few, but they did fail, we were able to rescue those failed and bring them back in by using the qualifier peptide. Because the peptides, even though there's a quantifier and qualifier, and I said, the best one you use is your quantifier, but usually they're pretty close. So we were able to do that. And there was still a few that we couldn't rescue, so we had to go to the second tip. So overall, unless you're doing this in the clinic where every measurement matters, if you're doing a large cohort, you probably can get away with a single peptide if you know your assay, right, and know the performance of the assay, which we do on this one. They've been used for a lot of different things. And again, I wanted to point out that this was actually when we did the whole cohort, which was all time points. There was just over 1,100 tips that we analyzed. And so we were actually measuring and quantifying 74,000 transitions for the whole data set, which was all of them, which is what we used for this analysis, where we're asking, really trying to help us understand how we should be proceeding forward as we go on to additional cohorts. So why was all of this important? And the first one is because we understood very early on that having the ability to use remote sampling devices across the board, you could start look at early intervention, chronic disease monitoring therapies, after hospitalization, marginalized group, as well as deploying it across the world. And we have worked with the Bill and Melinda Gates Foundation on dried blood spots. And we like that. And I'll show you one example of where we're using it at Cedars in a different country. Okay, so let's just talk about some examples. So this is one I really like, because this is on Takashima syndrome. It's a rare disease that happens in older women. It's often misdiagnosed. And so what we wanted to do, this was with, again, Noelle Berry-Mers. Essentially, you can enroll in this registry all online. So you can consent, upload all of your clinical data, including all your imaging, because this is hard. You always have imaging that goes with it. You are adjudicated, your status adjudicated at Cedars, blinded. If you have Takashima at some point, we will then send your remote sampling device in the mail. You then take your sample and mail it back to us. And what we wanted to understand is to build this registry out so that we could understand whether or not there was a mechanism underlying Takashima. So Takashima usually happens when there's incredibly high stress. You end up having a misbeating in your heart, and it looks like you're having a heart attack because you're not efficiently pumping out. But there's a very distinctive way that the heart contracts abnormally. And so we brought that in, and our controls were from a different cohort. It was done here. It's called the Corral study, which is healthcare worker study. And we had people taking both plasma and remote sampling devices at the same time, and they were declaring their health status using a questionnaire. So we did age match and sex match. But again, remember, this is primarily a female disease. And so we analyzed across those two cohorts. And what we found, it shows in the PCA plot in the bottom, is that there really was no overlap in the blood proteome of the individuals that were coming from being healthy, or healthy as they can say they are, versus those who had had Takashima, adjudicated Takashima event at some time in their life. The average mean year was average mean time to event had been 2 years, but it went out to as far as 10 years. And even then, at 10 years, their proteomes are still very different. And this is fascinating because everyone assumes that because with Takashima, if you survive it, it's an acute event, and you come back, everyone assumes that you're back to normal and that there has been no change. But clearly, that's not true, at least at the blood level. There's really quite distinctive differences. If you've had a Takashima event, 2 years or 10 years before that, you are still distinctive from a group of women who have not had that and who are declared healthy. Now we're going on and expanding this, and in this whole program, we also have a number of mice models where we can induce Takashima syndrome, and we're following both the blood samples of those mice as well as looking at the tissue, trying to understand what's going on, because this breaks a little bit of the common dogma that's out there saying that, once you recover, even though you're at a little bit more risk of having it again, you're really back to normal. And this implies, based on the blood proteome, that there's an inherent difference in systemic difference between these individuals who've had Takashima. So again, the idea then is if we can start to integrate and having the remote sampling devices really as more of a direct communication to the individuals, as opposed to having to go in and getting it done, which would in this case exclude those individuals. And it's very hard to track rare diseases. This way it was very easy. Consent's done, everything's done online, and then they do their sampling and we can send it back. So one of the other questions, of course, this raises is that that's great if you can use blood samples and remote sampling, but does it correlate to the more traditional plasma samples, right? And so we've done a number of studies to do that comparison. And this one's been published where we did 8 individuals and we were doing different types of blood collection. So using meter devices versus venous versus collecting plasma and then using blood or plasma. And then we analyzed it multiple different ways. We have another targeted panel. It's called Health Surveillance. It's larger. It has 27 FDA or LDT proteins being measured at the same time. And so we were able to do this study to really try to figure out what's going on and whether or not there was a good crossover between it. This is just a heat map of those proteins in the Health Surveillance assay. You can see that, they're not the same, right? Whether or not you're collecting, of course, capillary blood or venous blood or plasma, it's as expected, they're not the same. But the question is qualitatively, they are. So if we just pull out one of those proteins, so we're pulling out the data for CRP, well, here we're seeing it for every single protein. The concentration, of course, is at different levels, so they're different heights. But in the insert is actually the level for CRP. And essentially, the absolute values are different, but they're relative to each other is the same. So they are correlative to each other, at least for that protein. Of course, you have to do it for every protein that you care about. So that was good. And of course, the other take-home message, which has now been done in much larger cohorts, and we all know this, is that your blood proteome or your plasma proteome is more similar to yourself than it is to someone else if you're taking multiple time points. So you are distinctive to yourself depending on what's going on. So I want to just talk a little bit here about, again, where we needed to be able to look at plasma versus dried blood to see whether or not they're going to correlate. And this is a cardiac sudden arrest study. It's done with Sumit Chung here at Cedars. Cardiac sudden arrest is really when you have a sudden and unexpected cardiac arrest, right? And so it counts for a huge number of deaths per year, but it's in a population, which makes it very hard to study because you want to be able to predict who's going to have it, but yet that would mean you'd have to do population-wide screening, but it's not just one event because it can happen quite quickly. So you don't even know the timing to which you would want to do that. And then, of course, it's in a very small numbers, right? So it just causes lots of problems. But Samid has really a remarkable set of cohorts. One's called the Oregon SUD study, and the other one's Ventura Presto study, where they have been tracking using first responders to collect blood sample, not blood, plasma samples at the time of the cardiac arrest. And those that survive, they go back out and collect it. So they really are monitoring these very large populations of who's having sudden cardiac death or cardiac arrest, and then working through that. So we convinced them that maybe instead of collecting plasma, because there's a whole bunch of issues with how long it's going to go from when the first responder to actually getting it into a place where it's spun down to plasma. There's variation left at room temperature on ice, just very challenging handling of the sample due to the circumstances to which they have. These are remarkable that they have been able to convince the first responders in both Ventura and Portland to do this. This is just remarkable, but yet the analytics behind it are a bit tricky. So we suggested that we move to remote sampling devices because once the sample's taken, now there are things around this which also makes it challenging, right, because of the circumstance. But we've moved over to it. But that means that we need to be able to understand the relationship between plasma and meter devices. So we had already published on a plasma it's a great study, again, a remarkable samples. We have hundreds of these individuals that we analyzed on plasma, and we also did lipids to try to find a signature that might be indicated of an occurring sudden cardiac arrest. And there were, of course, coagulation, protein changes, not surprising, but there are also immune systems around IGF-1 and IGF-2, transport, and things like including the binding proteins that were changing in a way that was very good. So from that, and this is what we're currently doing now, and Jae-Hung Lee in our group has made an assay where we took those proteins of interest. There was 23 proteins begin, made a targeted assay and validated those targeted assay back on plasma, and then crossed it over to the meter devices and did a test case and validation study on this to see whether or not there would be proteins in this group that would be predictive of a sudden cardiac arrest and be present and increased in both or decreased in both plasma and meter devices. The answer to that is yes. So this is a AUC curve of the 7 proteins in plasma. We did it on 107 individuals, and then you can see on the replicate in the dried blood, we have a training set of 286 individuals, and then the test set of 122. And you can see for the AUC, sensitivity, specificity, and accuracy, it's pretty good. It's essentially going over. Is it perfect? No, but it's pretty good. We're very happy with the data across the board, and we're able to now flip between them. So this is a big step going forward. And these are just some examples of cohorts that we're analyzing. But I do want to spend the last few minutes because in the field of mass spectrometry-based plasma analysis and I'm going to show any body fluid, there's been a movement towards using nanoparticles to try to collapse the dynamic range so that you can see deeper into the proteome. It's always been, of course, a problem in plasma because you're dominated by IgG and albumin, and that just takes up such a bulk, right? And then you have everybody else. So we have worked hard for many, many years, decades, to get protocols that are very robust just to do naive or depleting the top 14 abundant proteins and then doing protein analysis. Probably our best, what we call for naive plasma, so untreated plasma, is to use SDS and then SP3 digestion, using trypsin non-digestion on an SP3 filter, and then analyzing that. And again, it's all automated on our big robots and able to do that. And we get probably for plasma anywhere between 800 and 1,500 proteins. It really does depend on the disease you're monitoring. generally speaking, healthy control, we see less proteins. When you have diseases, we see more proteins. And so what's happened in the last few years is that these nanoparticles, it's gone, different companies make different ones, but essentially allows you to collapse the proteome. So you do need to see bigger changes, generally speaking. So to, if you're in normal plasma, see a 5-fold change, you'll pick that up, obviously, using these nanoparticles, but maybe you won't pick up a 1.2 5-fold change, right? So there is a trade-off often, but you do get larger numbers. And that's been done in plasma. And I'll show you a little bit data from a company called Seer. And Seer, I would say, is the leading company in this field. They have set the gold standard, but they are not inexpensive. And so what we wanted to be able to do with the dried blood is to use the same concept for nanoparticles, but because we'd have so many more samples, because you're now not doing a single blood draw, you're doing multiple blood draws, we needed to find something that was cost effective. And so we started to look around for other nanoparticles. I'm going to talk about a company from Ceres that has a set of nanoparticles that we're in collaboration, and this will actually be the first time we're publicly speaking about it. And this work was done by Amanda Campbell. She was a visiting postdoctoral fellow, graduate student. And then we have Lindsay Becker and Kuhn, who have been leading it. And this was done under our precision health initiatives. So let's take a look at it. Okay, so this is complicated, but it's not. So what we're going to compare here is, in gold is Seer. This is plasma. If we take plasma and use Seer nanoparticle, we go from 1,000 proteins up to almost 6,000 proteins. And if you have a disease, you'll get up to 8,000 proteins that you can measure. And that's in the gold. In the green is our old method, our naive sample, right? Where we're going through SDS plasma. We're getting around 1,000 proteins, right? Big difference between the nanoparticles now. So what you're seeing in blue and red are samples using 2 different buffers, using the series beads. And you can see that they're getting close, but they do not reach that of Seer, but they're getting really great protein identification. And below what you see in the whisper plots, or the violin plots, is the coefficient of variance. The dotted line is 20%, and you can see Seer is an incredibly tight coefficient of variance for all the proteins it's measuring. And the first 2 blue are representing day 1 and day 2. So these are 2-day replicate total protein workflow replicates, and you can see there's pretty good. The intraday is the third one. So the third blue and the third red are the intraday between it. You can see still pretty good. Most of the proteins are hovering around 20% coefficient of variance. But the graph on the right, I really love. This is actually, I think, tells you everything. So the percent CV is along the x-axis, the y-axis is the cumulative protein numbers, right? So you can see gold, it comes up, and if you just went down 20% or less, you can see 80% of the proteins that we're measuring in Seer have a coefficient of variance under 20%. And as you go down at the bottom, you can see in green, our former one, really, you can get up to 80%, but you're now about 50% coefficient of variance, right? So this is not, it's good, but you get there, and that's just how it is, right? You just have to have just know which peptides and which proteins have that kind of coefficient. But you can see from the series set that they also do quite well, right? You can at 20% get up to between 60 and 80% of the proteins having a coefficient variance of under 20%. Of course, you want it to be down at 10%, but there is a reality check. But that was what we did to get to that. We also tested things like digestion efficiency. We always have to do these things. And of course, there's inter and interday variability that we have to worry about. We also looked at extraction, because these are new nanoparticles. We have to do all of that analytics to really try to optimize the workflow. And this is done on a plate basis, not up on our i7 yet. We're still doing it manually on the 96-well plate. We're just about to we're working with Beckman Coulter to get it up on our i7. So I think the coefficient of variance will be better, but it's still incredibly good. So this was, we settled on the 2-hour, we did 2, 4, and 6-hour digestion is the data I'm showing you here. We get it just over 20. This is healthy and control samples. As I said, it's always lower. So it's just around 2,500 proteins that we're quantifying. And you can see essentially the longer we go out, it doesn't really change the number of proteins we see, nor does it really change the coefficient of variance, which is shown on the furthest right-hand side. So our optimized method is now 2 hours. We can probably go down faster if we want, but that is a day workflow. So we want to be able to do two sets of samples per day on our automation system, and the 2-hour digestion works. And like I said, we could probably move it down even lower. Protein numbers are a little bit lower, but really marginally. And again, looking at the cumulative protein numbers versus coefficient of variance, the 2 hours actually for all those proteins we're measuring give us actually the best coefficient of variance. And that's actually not unexpected. That's well known about digestion. If you over digest, you start to lose different things happen. So you over digest. So this is now looking at patient. This is a patient with Alzheimer's disease. And what you can see is really that we get beautiful depth. We're getting great cover. This is now the same optimized method over 3 days. So looking for each day, we're doing it 3 times, and then 3 times over 3 days to look at efficiency, to look at inter- and inter-day variability. And it's beautiful. It's really highly consistent across time. And so that's a really robust assay. The proteins that we see that are unique to the dried blood when we're doing nanoparticles versus plasma, not surprisingly, we just get a really nice increased diversity as you get deeper into the blood proteome. Okay, so I'm just going to finish this with a cohort that we're excited with. This is the first one we're using nanoparticles on. This is a cohort coming out of our surgery here at Cedars-Sinai. It is looking at rheumatic fever valve disease. And of course, if you get rheumatic fever, what happens is you end up having or can have valve, and you have to come in and get your valve replaced. So there's been this great collaboration with the surgery department in Zambia, where they've been trained at Cedars. They go back and start to perform the valve replacement there. So we wanted to be able to, because rheumatic fever rarely happens in the United States, we will still want to be able to measure it in a place where it happens, and to be able to measure them as they come in, get the surgery done, and then leave, and then we can track them wherever they are within the country. And so remote sampling devices seemed the logic to do it. So this is our first cohort, 120 individuals, 3 groups. So it's controlled, had rheumatic fever, but no symptoms right now, had rheumatic fever without valve disease. So controlled just healthy individuals, those that have had rheumatic fever without valve disease, and those which have valve disease, right? And so this is called the Dream Care cohort. And this is just showing you the quality of the data. So we have the 3 groups. The majority we're detecting using the series nanoparticles on this. We're getting just over 3,000 protein quantified. You can see that there is differences between them, but they're not uniquely different. But the best data you can see essentially in the total protein quantities. You can see they're all hovering for every single sample about the same. And that actually transforms to about what we call missingness data. So the data that we're not obtaining, you want to have that almost be 0. So when you look at the bottom left, you want to have that completely black, which essentially is what we have. So this is giving us really consistent data across the board on all these individuals. And you can see again the dynamic range that we're reaching. It's really beautiful. So again, we've been implementing remote sampling devices. I've given you examples of early intervention, monitoring, after hospitalization, trying to go after marginalized group, and just starting to deploy them at locations around the world. So we're really excited about the work. But behind that, took years to be able to make sure that the analytical performance of the workflows were good. And of course, a huge number of people who have worked on it over the years, everything from the automation to now the enrichment using different nanoparticles. We have these great relationships with the companies we work with, and a really remarkable group of clinicians that we also get to work with. So thank you very much.
Thank you, Jenny. Really, really interesting talk, and lots of data, lots and lots of data. Brilliant. Please, for the audience, if you've got any questions for Jenny, please could you post them into the chat? It'd be great to get your questions answered from Jenny. I have a couple of questions to get us started with, Jenny. You kind of hinted at it a little bit at the end there, but just wondered whether you could talk to us a little bit about how much these assays are now being deployed in your hospital, but also elsewhere beyond your laboratory, beyond your hospital. Are they becoming routine use, or is this still a very much experimental?
Unfortunately, it's still experimental. I would say it's in really the world of research and epidemiology, right? I think it has not yet made it to the clinic. And I think there's multiple reasons for that. One is it's mass spectrometry based, which means that would have to get into the clinical chemistry more for proteins, more so than it is now. Two, and with that, we don't have full menus yet to make it, the cost effectiveness that's required for assays like that. Number two, it's hasn't been done because, if you're in the hospital, it's easy to get the plasmas. It really has to be doing for those times once someone leaves. And there's lots of cases where that would be good. Following heart surgery, being able to monitor an individual, rheumatic fever, being able to follow in many places where this could be implemented, right? But it hasn't made it. And again, that's also partly because the markers aren't there, right? The assays that you would want to do, other than things like CRP, coagulation, there are like, at least I think that we can measure quantitatively with great CVs. There's probably 40 clinical assays that are already there, but people are not using it. So the whole field of how you use multiplexes, how you use mass spectrometry is still evolving, I would say. It's getting very close. So, but once we do it, it's a great way to do it because you're going to have lots more samples coming in, right? Not just a plasma sample. You're going to have maybe 10 remote sampling devices coming in for that same way. So there is definitely a business case there, but it's not implemented yet into the clinical chemistry lab for multiple reasons.
Yeah. Is there any particular thing that could be done to help change that, to make it more implemented, Janet?
Yeah, so I think there's getting people used to thinking about using remote sampling devices, even epidemiologists who that's just their trade. A lot of them don't know about them. And I just, I sit on the UK Biobank External SAB board, and it's like, this should be implemented because it's a great way to track cohorts like that are population based. It's not even there yet, right? And we have it going into one clinical trial now where it's multi-centered. It's really a beautiful trial looking at TRAVR, which is, again, a valve surgery. And we're going to be monitoring individuals because we know that sometimes it doesn't work very quickly within a few months, and we need to get them back in and do it. So this would be a way to follow individuals. So it's getting out there. But again, you have to have the clinical case, right? You have to uptake to do it, the acceptance of that. You have to have the assay that's going to be used, right? You have to figure out your CPT code. So there's many things. So how do you help that? The mass spectrometry companies have to help. The clinical chemists, and there's a lot of people in the clinical chemistry world trying to get protein mass spectrometry into place. And there has to be a business model that helps biomarkers move into the clinic, which has always been hard to do that. We don't have a lot of new markers coming through. And there has to be an acceptance of multiplexing, and there has to be a cost recovery basis that do it. So I think a lot of what this group does and advocating for the use of remote sampling will help. But I think there's a lot of barriers from a lot of different groups have to get involved.
Yeah. Thank you, Jay. And just mentioning on that, just a little plug for a PCSIG event in December in Florida. We've got a workshop which is advertised on our website, which is going to be looking into this. How do we move this from the academic, important, interesting into the mainstream of clinical diagnostics that's available, and actually starts to make a difference to a lot of people's lives, which
Absolutely.
I think it can. So I encourage people to take a look at that and maybe register to come along to our workshop, University of Florida, 1st to 3rd of December later this year.
Excellent. Yeah, I think these things are very important, and adopting them into clinical trials, expanding who can participate, because now you can do it from wherever. So there's lots of really compelling questions that can be answered, and Yeah, I think, I think mass spectrometry is a great way to measure from these as opposed to immunoassays because we can digest it. It doesn't matter which methodology, if it's dried, because we don't have to worry about refolding correctly when you're using an antibody-based. So there's lots of reasons to do mass spectrometry, and people sometimes get scared of the technology. They shouldn't be. It's a great technology.
Absolutely, Jenny. So we've got a question here from Ian. Thank you, Ian. How much improvement would you expect to see when you automate the Ceres nanoparticles?
Yeah, that's a great question. So right now it's plate-based, and we all know pipetting's pipetting. Yeah, so I would expect to see the numbers will not change. I don't think we're going to see more protein, but I do think the coefficient of variance will drop in half. So it's probably going to get down from where it's hovering now at 20% for, let's say, 80% of the proteins. I think we'll probably get that down to 80% of the proteins under 10% coefficient of variance.
Okay, that sounds cool. Again, everyone, go on, sorry, Jenny.
But we'll see, right? You have to do the experiment.
Yeah, we're scientists. Hopefully that answered your question, Ian. We've got a couple of minutes for anyone that wants to submit any further questions. It would be great for Jenny to have that. Oh, Ian just came with another one. He was obviously busy typing away. Have you done any multi-omic analysis on these cohorts to assess if the proteomic data orthogonally compares to the transcriptomic data? And if those pan out, do you think it would help translation to the clinic?
That's a great question. So my favorite multi-omic study that we've done is called Molecular Twin. It's a framework to which you can do, it was done in pancreatic cancer. And there essentially we threw every omic at these cancer individuals. So we had plasma and we also had tissue. So we did genetics, we did transcriptomics, we did lipids, and we did, and against tissue as well as, and what we wanted to know was we did both survival and reoccurrence. So again, the trick to that study is you've already been diagnosed with pancreatic cancer and trying to do prediction of your outcome. That's very different than talking about early detection where we know genetics plays a big role. And in that data set, essentially genetics had no bearing at all on outcome or your survival. But again, they already know it, right? So a lot of the impact of genetics is, do you get the disease or not? So the prediction going forward, it was not helpful. Transcriptomics of tissue was okay. But what really dominated, and then we validated it in two other cohorts, was actually the plasma proteome. It was by far the most important predictor of all of the omics. And not to say that the proteome and transcriptomics of tissue wasn't important. They did and could contribute, but the plasma proteome alone had AUCs of like 0.92. So the other ones could boost it up a little bit, but almost not at all. And so the dominance there, I think, was quite surprising, but maybe not, right? Because again, what you're asking is, you have the disease, what is your response to the therapy? Are you going to survive? Are you going to have reoccurrence? And that is a very active one. So I would have predicted transcriptomics and proteomics of tissue would be the most important. It was wrong. It's more of a systemic, but again, that's cancer. So there's lots of things around it. So I think depending on the disease and the drivers of the disease and the question you're asking will dictate which of the omics is most important or which combination of omics is more important, right? To outcome, to diagnosis, to prediction of prevalence and those types of things. So I think, will it help if you had a genomic component to a biomarker? It might make it an easier sell, especially in places where they're used to doing that, like in the cancer domain. In other diseases like IBD or cardiovascular, where there is a genetic component, but maybe not a dominance of it, I don't know, right? I think proteins become equally important because you could say there's a lot of diagnostics around proteins already, right? In cardiac, we do TNI, BNB, CRP all the time, right? So again, I think, you kind of have to think it out, but I could see how other omics can help, but it does make it more complicated and harder to physically get into a clinical lab.
Thank you, Jenny. And it turned out there were two different Ians that asked the questions. What were the chances of that? It looks like we've not got any other questions. I have one last question for you, Jenny. how much are you, as part of this, in the design of your studies and your experiments, how much are you involving patients to
Yeah,
co-design this with you?
yeah, it's a great question. So we involve them, of course, during the consenting and getting them to do it. And I think there's a lot longer relationship than when you just do a plasma draw, right? Because you get a plasma draw. But because this is remote sampling and you're doing it as a continuous, trying to do it as a continuous harvest of the samples, then I think there is a better relationship. What we have not done is we have not gone back, except for at the very beginning, ask them, so how did you find this? Does it work or not work? At the beginning, we did that a lot to try to make sure that we could fine tune our interactions with our patients and the individuals involved in these studies. But probably that's not, we probably should do it more, right? To try to continue to improve it. We have great compliance and always have had great compliance. So maybe that's one reason we haven't. But I do think as we start to move more into information right now, most of this is still at the discovery level, trying to find those biomarkers that are trying to understand the mechanisms that are driving the flow and fall of the diseases, trying to get so we can actually see more of the proteome. I think as we move forward into some of these new studies that are starting now, that are more interfacing with the participants, then we need to be figuring out how to do the feedback.
Yeah, thank you. Just for you and for the audience, just for information, on the PCSIG website, we do have some templated patient questionnaires that you can use to help get feedback from your patients. And we also have some materials on patient public involvement, getting them involved right at the beginning in the design of the protocol. Not just asking their opinion, how did it go, but how should we do this study, which is becoming a very important thing for healthcare, because we're talking about patient-centric healthcare here. So we should be designing the studies with the patient, not for the patient, kind of thing. And I think it's a subtlety that we're all learning about.
I think, Neil, you just said that very elegantly, and I heard.
I think it's for all of us to learn for patient centricity. Anyway, Jenny, this has been an absolutely wonderful webinar. I've loved it. I've learned a lot. Just like to say thank you to you and to the whole audience out there. And thank you to Ian and Ian for their questions. For those of you not members of the Patient-Centric Sampling Interest Group, if you want to learn more about us, the QR code on the left there will take you to our website. The QR code on the right will take you to our LinkedIn group. It's all free. All of our content is free. Please join the LinkedIn group, ask questions, share your information. It's there for us all to help move forward this field of patient-centric sampling. And we've got some upcoming webinars. We have one a month. We've got we're booked out till late 2027 now for our webinars, but we've got two coming up in June. One, improving access to blood tests for people with learning disabilities. Really exciting one. That was a follow-up to a webinar we had before, and a film that we helped make that's had such a huge impact with patient groups. We're having to follow up and see what we can do to help move that forwards. And then Chiara is going to be talking to us about remote monitoring of anti-seizure medications at home through microsampling devices in epilepsy, and that's later on in June. So we've got a busy June for webinars and a busy time coming up. I'd like to thank all of our sponsors again, without whom we couldn't do all of this. I'd like to thank you again, Jenny, and all of the audience today. So thank you all. The recording of this will be on our website and on our YouTube channel quite soon. Thank you. Goodbye all.
Thanks, everyone.